There we are. So, as Steve said, I'm Katherine Frels. I'm the small grains breeder, and I'm also one of the science leads for the Coalition for Grain Fiber. And so I wanted to really highlight that we started with wheat just because that's where we had a mass of breeders and researchers really interested in this project. We really believe that we want to expand this project beyond just wheat. We want to, consider other fibers, other benefits that we can bring. We have a great mass of people here that are interested in developing healthier food, making it easy for people to be healthier, just like Rod said. So this is a paper that I found saying, revisiting the contribution of maize and wheat to human health and nutrition. And basically it says that we know how to get these essential nutrients in crops. Why aren't we doing it? It goes back to your question earlier. Why haven't we done this before? It's because we really weren't thinking about it. We were thinking about medicines, not food as medicine. It doesn't include rice in this particular article, but we are here today to make the argument that rice is just as important as wheat and maize in making these bioactives, dietary fiber, minerals, reducing anti-nutrients. That's just as important in all of these species, and we need to make an effort. And I wanted to highlight the last bit here. It says agri-nutrition studies should be multidisciplinary and whole food systems oriented. What are we here at the Coalition for Grain Fiber? We are multidisciplinary and whole food system oriented. So we are well positioned to address this. So starting with wheat, we know wheat produces most of it, a large portion of the calories and the protein and the dietary fiber that the world eats. We also know it's a significant source of mineral nutrition. What I'm going to talk about for a short time today is just fiber and nutrition. I want to share some thoughts we have on kind of a phase 2 for additional target fibers. This is not an expansive list. This is just a quick look at what we could do. And also, how could we combine high fiber wheat with micronutrient biofortification efforts? This is something that has come up from the very beginning with wheat breeders is, okay, if we're going to do this for fiber, why can't we combine with other biofortification efforts to really improve U.S. wheat? All right, so we have focused our CGF 1.0 on water extractable arabinoxylans, a little bit on resistant starch, but that's leaving out a whole other component of arabinoxylans. This is work from Devin Rose's lab and is really highlighting a Kansas breeding line right here that is high in total arabinoxylans. So we know that it likely has more water unextractable arabinoxylans, which are insoluble dietary fibers. So compared to the solubility in the water extractable, these unextractable arabinoxylans have the potential to improve gut motility, function, also potentially absorption of undesirable materials. We don't have funding to do this yet, but we started saying, okay, wheat breeders, watch for things that are unusual in your breeding lines. Look for lines that are high in water absorption, have higher than expected water absorption, but aren't high in protein, aren't high in these other traits that could contribute to water absorption. Alan Fritz down in Kansas said, hey, I've got one. And it happened to be in the regional nurseries that year. So I gave some extra seed to Devin, and sure enough, what do we find? It is high in total arabinoxylans, higher than UMI 34 and Valoris, those first two lines that we're working with. However, it's lower in those water extractable arabinoxylans. This is a high yielding line, variety release candidate. has been running around our regional nurseries for a while. So we know we can find these high fiber wheats in the US in our elite materials. This one has a complementary dietary fiber profile that we could stack potentially with those profiles from Valoris and UMI 34. We don't know what's causing this yet. We have actually proposed a QTL identification project, or basically determining what DNA marker is associated with this trait. We just resubmitted that grant in October to really facilitate rapid marker-assisted breeding and discovering what's going on in this breeding line. And Brett's here today. I did not put this slide in there for you, Brett, but Brett has talked about beta-glucans for a long time. This is another fiber that we really need to be considering in wheat. Wheat is not high in beta-glucans compared to barley and oats, which actually carry heart-healthy labels. Wouldn't that be awesome if we could put that heart-healthy label on a wheat product? If you're not familiar with beta-glucan, it has that heart-healthy product label because it's reducing cholesterol, lowering glycemic index. That's why we're all told to eat oats for breakfast and not wheat products for breakfast. Let's change that. It is primarily found in that allurone layer. So, you know, you can get higher beta-glucans in whole wheat products, but we need to get it in those white wheat products. You know, is that something we can do with genetics? There are studies here that show we can mine genetic diversity from wild wheat relatives. We really don't have a lot of genetic diversity in domesticated bred wheat or even really in domesticated tetraploid wheat. But our wild species, a little bit in triticum, but in that age-elapsed species, we actually have a fairly large beta-glucan range. And we have projects ongoing led by the USDA and the Wheat Genetics Resource Center that is already looking to get these wild relative genetics into domesticated wheat. And those are panels that we should be looking for beta-glucan content in. So we could try to stack those beta-glucan QTLs with AX QTLs. We have several panels that have identified some quantitative trait loci. We can leverage that and include that in our grain fiber 2.0. So what are we talking about? We're talking about that fiber gap where we can, whoops, let me go back, where we can get with our current targets, our current genetics available for high fiber wheat. We're talking about that potential 25% reduction in a fiber gap. As we keep stacking those additional fibers or finding different genetic diversity that improves fiber over the genes we have right now, we're potentially looking to really target a 50% reduction, getting that white flour to, you know, much closer to today's whole wheat flour. Again, getting that white flour product that people prefer to consume closer to that whole wheat product. So I wanted to end today, move on from the fiber, because again, Steve said we don't want to be a one-trick pony. How do we really work on that healthier wheat products? Well, we could look at vitamins, minerals. So I wanted to highlight some mineral research that has been done in the wheat community. There's been multiple studies looking at biofortification efforts of zinc and iron, as well as a little bit in selenium. As far as I'm aware, most efforts right now that are available in wheat lines are related to zinc. The Harvest Plus project has really focused on zinc, but iron is a huge need. I'm just back from maternity leave, so I know that especially for women, the nutrient requirements are so important. Iron and zinc. Guess what? I passed my glucose test while pregnant. at the same time, they said, oh, actually, you're anemic, so you got to start iron supplements. Wouldn't it be great if I didn't have to take a pill and I could just eat more bread? So we know we can do this. We could target other minerals such as selenium, copper. We know that wild wheat relatives are key sources. Again, that's those same panels that we're using for other traits. We really need to be mining them and bringing them into CGF. We can make wheat a superfood. There's a caveat though. When we increase other minerals, we risk increasing minerals that we don't want, anti-nutritive things. Cadmium is a heavy metal, and the data shows that grains including wheat, rice, maize, contribute 25% of human cadmium consumption. Again, things that I worry about. Pregnancy increases absorption. It is a regulated anti-nutritive consideration in the European Union, China, Australia, and I believe the US is looking at regulating it more. I can't remember if it's just in infant food or in food in general, but there's a lot of conversations going on. We talked about it in Wheat Quality Council the last couple of years that cadmium is likely to become regulated in the US. And we actually here at Nebraska are well positioned to study this because we identified that we can test for cadmium content in grain here in Nebraska. We have high cadmium soils. So what else did we discover? Well, this paper also from Mary Gutierrez showed that cadmium is retained at a higher rate in the milling process than iron and zinc. So if we have high cadmium varieties that are also maybe high in iron and zinc, we're more likely to retain that cadmium in white flour than we are to retain high levels of iron and zinc. This is why it's a concern. We know that we can handle this, but we have to be looking for this cadmium concentration or other heavy metals. What we also showed in this study is that we can select wheat with decreased cadmium concentration while maintaining other higher minerals. So we showed in this study that zinc and cadmium were independently controlled, meaning that we can select high zinc, select low cadmium. But again, the key message here is that we actually have to look for these minerals. We have to be monitoring our breeding lines. Otherwise, we're not going to find that. And that's something that we can do in CGF. We can be a group that says, hey, we want to improve the mineral nutrition in wheat, but we have to provide funds, help find resources for breeders to actually go looking for them. Devin and others in this room can vouch for it is not easy to measure these. We don't have a really fast way to measure fiber content for a breeder, and we definitely don't have a fast way to measure mineral nutrition in our wheat varieties. So that's something that we really need to think as a group. If we want to have these goals, how do we make it feasible for breeders to do that? So I'm going to wrap up there and just say that we have built this coalition in just a few years into a fairly large coalition. Nebraska has really been on the leading edge. We have collaboration initiative funds from the university. Nebraska Wheat has provided really a lot of the kickstarting. We had a conference grant from USDA NIFA that helped support some of really the first roundtable that we had. And then our whole Coalition for Grain Fiber group. And I'm really excited to see this grow. I'm excited to see those next steps that we can take beyond the first set of fiber materials.